So welcome everyone to the GW JDD Dermatology Translational Science Lecture Series. I'd first like to thank our partnership with Orthodermatologic to help support this program. And now it's my absolute honor and privilege to introduce our invited speaker, Dr. Angela Christiano, who is the Richard and Mildred Rodebeck Professor and Vice Chair of Research at Columbia University in the Department of Dermatology. Unless you're like a survivalist somewhere in the wilderness with no access to internet, TV, phone, you've heard about her work. Dr. Christiano has been central to our understanding and translational development of new therapies for immunologic-mediated alopecias like alopecia areata. I think she's won possibly every basic science award that is possible in dermatology, but there's so much more. She served in multiple positions. She was past president of the Society of Investigative Dermatology, deputy editor of Journal Investigative Dermatology, and she's also an entrepreneur, which was an element of derm that we really don't get a lot of in our training. And she's actually taking her bench research and literally translating it to the bedside. So it is my privilege to welcome Dr. Christiano.
That's great. Thank you so much. I always feel so old after hearing those introductions. Thank you, Adam. It's such a pleasure to be here and see so many old friends and new friends, and this convergence of New Yorkers in Washington. It's wonderful to be here. Thank you for having me. I'm going to try and, I have, 45 minutes today? Okay, so this will be a walk through some of our early work in understanding a bit about the genetics, and unfortunately there's a little immunology in the middle, but if you can stay awake through that, I promise the rest will be fun and easy. But I always start with this slide because it reminds me to acknowledge my clinical colleagues. So as a PhD basic scientist, I'm completely reliant on the field of clinicians to help anytime we embark on a study like this. So I have two colleagues at Columbia. Both of them have now moved on, but Julian used to run our clinical trials unit. Some of her former fellows are here. And Raphael Kleins was a MD PhD immunologist who joined the project halfway through. And most of the work that you'll see early on really rested on the shoulders of the Alopecia Areata registry, which was a 5-site collaboration between Columbia and these sites. And each of these PIs were instrumental in both educating me about alopecia areata and also in collecting the samples that were necessary to do this work. These are my disclosures, and it's probably important to point out that my collaboration with Aclaris is on JAK inhibitors, but we won't be talking about their compounds today, except at the end, but it's in mice. So this is an introductory slide that to this room needs no introduction, except to say that alopecia areata comes in many forms and many flavors. When we started working in this area roughly 20 or so years ago now, these sorts of pictures would appear in the literature. You all know this as a swarm of bees pathology, where you see this infiltrate of immune cells surrounding a growing hair follicle. And for a long time, the composition of that infiltrate wasn't very well understood. And so some of our work has been focused on identifying the nature of the infiltrate, and in particular, whether there were cells within that infiltrate that were the disease-causing cells. But just to, again, in disclosure, I hadn't thought much about alopecia areata either. When I was a fellow at Jefferson, I worked on epidermolysis bullosa as my postdoc. So I was already in the field of dermatology, but hadn't really thought at all about hair. Then I moved to Columbia in 1995, and a few months after I got there, this is me, so my own hair fell out. I developed alopecia areata as a patient for the first time, and was met with the same thing that we're pretty much stopping to tell patients, which is we don't know what causes it. We can't tell you if it's going to get better, worse, or stay the same, and there's not much we can do except give you steroids. So at least the third point is getting a little bit better. So I hope by the end of today I can show you kind of the evolution of the work, how we came to study this, the approaches that we took early on, and then finally, at the end, the last larger part of the talk will be some of the clinical work on JAK inhibitors. And if we have time at the very, very end, there's some new unpublished data that I've been sharing in the past couple weeks or months, just for reaction from the crowd. So be gentle if we get that far. So the concepts for today really focus around this model of the hair follicle normally being a site of relative immune privilege in the body, which means that if all's going well, the hair follicle is cloaked in molecules that Ralph Paus calls guardians of hair follicle immune privilege, meaning that they shield the hair follicle from being recognized by the immune system. For reasons we are beginning to understand, this type of immune privilege can collapse, and these deflector shields can go down, and the hair follicle can become vulnerable to recognition by these immune cells. And when that happens, it sets up a vicious cycle so that the hair follicle begins to secrete molecules of danger that say to the immune cells, come in and kill me. The immune cells then come in and get more and more activated as this process continues. And so this, again, is a bit of a vicious cycle, and finding a way to break this therapeutically is really what we'd like to aim for. So even though it's complex to look at, the goals of therapy are going to be twofold, and you'll see these concepts coming up. One is, if we administer a drug, we want to see clearance of the immune infiltrate from the skin. So we're going to talk about how to measure it at first during disease, and then when we're having a good therapeutic response, how it looks when it's cleared. And the second is, are we restoring the hair follicle back to its state of relative immune privilege? So if we're doing our job therapeutically, those are the two things that we're going to measure in different ways, by histology, by flow sorting, and then also by RNA sequencing. And so we'll come back to that. So the overall approach is something that we as geneticists call functional genomics, which is a great buzzword, but what it really means is, if we make a good unbiased map of a disease by starting with no preconceived notions, can we use that new information to lead us through the process of picking perhaps repurposed drugs that were developed for other diseases and applying them to alopecia areata. And the key to that, again, as pure geneticists, is making that unbiased map. So the field of alopecia areata had come with a lot of biases. What it might look like psoriasis or atopic dermatitis. And so really figuring out how to do the genetics correctly was, I think, one of the first steps that sort of broke open a new way of thinking. So again, we'll touch mostly on the translational aspects, but in order to get there, I think it's valuable to just understand a few slides about the genetics and then a bit about the immunology before we dive into the fun part. So this is the summary of, 10, 12 years of work on the part of the whole registry and a graduate student, Lynn Petkova, who devoted her PhD to this work. But this is called a Manhattan plot, not because of being New Yorkers or that this was done at Columbia, but because this is supposed to look like the chimneys on top of skyscrapers. So the red line here is statistical significance. So everything below that line is noise. Everything that rises above the line are locations in the genome where there's a suggestion that there's a genetic influence that's leading to susceptibility to alopecia areata. So we know that the HLA should be involved because we think this is an autoimmune disease. But the rest of these are basically the new information. The ones in black are sort of what we think of as the common core of autoimmune disease genes. So these are genes that are involved in not just alopecia, but many different autoimmune diseases. But the ones that we look for as geneticists, the ones that really get us excited, are the smoking guns, the genes that are implicated in your disease and your disease only, and no other disease, because usually those are the ones that have the highest value as targets because they tend to be more specific. So the one that you see in red here, with a horrible name, ULBP3 or 6, fits that criterion. It's a gene that's only been implicated in alopecia still. It's our second most significant genetic hit, which means it's low-hanging fruit, meaning a lot of patients carry genetic hits at this locus. But more interestingly for us is the category that it falls into. It's a danger signal. So an NKG2D ligand is a gene that is usually off in the normal situation. It gets turned on under conditions such as cancer, infection, stress of the cell, and it's basically a kill me signal. So when an NKG2D ligand goes up on a cell, it labels that cell for destruction, and then a killer T cell can come in, kill that damaged cell, and usually get rid of whatever it is, the infection or the tumor. In autoimmunity, what happens is it gets stuck in overdrive. So the NKG2D ligand goes on and it stays on. It doesn't seem to be able to come back off, labeling those cells permanently for repeated destruction. And we'll talk more about that. The other sort of main eye-opening change was, in addition to finding out that NKG2D as itself and an axis was important, was the realignment of alopecia areata with unexpected partners. So again, we went into this thinking it's going to resemble psoriasis, vitiligo, atopic dermatitis. But in fact, if you look down these columns, you'll see that by far the closer alignments are type 1 diabetes, rheumatoid arthritis, and celiac disease. This was a big surprise for us and took us many months to get our heads around because it was so unexpected. Really, no screaming skin phenotypes in any of these, and it wasn't totally clear to us until we really started digging through the literature. And we'll talk a little bit about that, about what might be common. But this was critical. That re-examination was essential, because without that, I think we may have continued to go down the wrong path, thinking that the immunology of alopecia was in fact more closely linked to other skin diseases. So I hope I can convince you now how we've taken those initial genetic clues and begun to use them to, again, sort of, now that we have a thread, you can sort of pull on the ball of wool a little bit and begin to untangle a bit of a message. So one of the main keys to understanding this came from, again, the exhaustive search of the literature to say, what is it that we share with these other diseases? And if you look carefully through the old literature, in all of the other diseases, there's a similar overexpression of a death signal of an NK ligand on the target organ in each of these conditions. So in rheumatoid arthritis, the synoviocytes do this. In celiac disease, the gut epithelium does it, and the islet cells, at least in type 1 diabetes mice. The nomenclature is horrible, so the NKG2D ligands have very bad names as they travel through mouse and human, but RAE is the mouse equivalent of human ULBP3. So our question really was, do we see the same thing in an alopecic hair follicle? So this is a control hair follicle. The blue is just DAPI labeling of nuclei. The red staining is a little bit of danger. That's normal levels of ULBPs that are present in the dermal papilla. But if we look in lesional skin of alopecia areata patients, you can see this bright red staining all along the outside of the hair follicle. So that's the dermal sheath layer. And it puts the label right in the right time and right place, if you will, to attract immune cells, because it's basically the outermost layer of the follicle before it hits the dermis. So we saw this reproducibly in many, many patients with alopecia areata, and it was a quite striking finding. The logic would say that if we have the ligand on the hair follicle, we should have the corresponding receptor on those incoming T cells. And so when we look at the immune infiltrate, you can see that NKG2D, which is the receptor for this ligand, is present on many of the infiltrating T cells, as is the co-receptor CD8. This qualifies these cells as killer or cytotoxic T cells of the CD8 phenotype that are attracted to this target by virtue of the fact that it's labeled in a kill me signal. So it was the first indication really that there must be an identity to some of the cells in this infiltrate that could give us insight about selective targeting. Maybe it's these cells, and potentially only these cells, that need to be targeted for therapy. This is the hard part, so if you want to sleep, now's a good time. So take a nap. So once we had that clue, it became important for us to go back a bit and to try and revisit some of the lessons that had been sort of brought forward in alopecia areata to see how we might be able to reframe the thinking a bit in light of these findings. So we were very lucky that John Sundberg and his colleagues at the Jackson Labs had spent many years developing a mouse model for alopecia areata. So this is an inbred strain of mice with a horrible name, C3HHEJ. It's not a transgenic or a knockout. It's a simple inbred strain. And if you leave the mice to themselves, about 10 or 15% of females will develop alopecia areata spontaneously in about a year, which is a horrible model for us because we can't sit around for a year waiting for the mice to lose their hair and not knowing which mouse to treat. So fortunately for us, John and his colleagues developed a transfer model where they could take a bit of skin from a mouse who's lost its hair and move it onto a recipient C3H, and then the mouse will lose its hair in 6 weeks. So I'll show you how we've been able to use that model. But to convince ourselves that the model was really a reflection of what we were learning about human alopecia, we needed to do a little bit of work. So this is just a quick gene expression comparison between human AA and mouse. If you look down this column, you'll see signatures that are quite similar. So many of the same genes are dysregulated in mouse and human AA. You're also beginning to see the beginnings of a signature. So these two genes are interferon gamma response genes. This one, CD8 and granzyme, those are signatures of those cytotoxic T cells. So even in these early studies, we begin to see some of the character of the infiltrate coming through here. So good correlation on the gene expression level. Do we see upregulation of a death signal in the mouse? So here's our human kill me signal, the NKG2D ligand. This one in the mouse has, again, another name called H60. This is a normal black 6 mouse, and you see very little red staining here, as we expect. This is a C3H mouse that does not have alopecia yet. And you see a little bit more staining even at baseline. It looks like it may be primed for activation. This is a C3H mouse that does have alopecia, and you can see that bright red staining is even more pronounced. So at the level of the kill me signal, again, good correlation between mouse and human. And then finally, do we see the infiltrate? So again, here's our human killers co-labeled with CD8 and NKG2D. Do we see the same in the mouse? Yes, we do. Here's a very nice co-staining of the same two receptors on mouse infiltrating T cells. But with mice, we can do a lot more. And so this is exciting for us because we can take all of the T cells out of the skin, and we can measure how many of those T cells are actually of this type, of this killer type. And you can see here in alopecic skin compared to non-alopecic skin, a full 20% of the T cells in the skin of these mice are killer T cells, compared to less than 1% in controls. That's a huge number. That's buckets of T cells. And so what it means, though, therapeutically, is it gives us something to measure. If we can clear this 20% from the skin and get it back down to 1% or less, there's our therapeutic window. So we don't have to kill every immune cell in a 10-block radius. If we can just get in and kill this 20%, we may have a chance at selectivity. This is the experiment that supports that. So this, for scientists, is the critical, necessary, and sufficient. Is it those cells and only those cells that are conferring disease? So this is how we do that. So we take T cells from an affected mouse, and first we inject all of the T cells into a recipient, and we show that the recipient gets alopecia as we expect. But then we split out our killers here that are positive for NKG2D versus all the rest of the T cells. When we inject all the rest of the T cells, nothing happens. The mouse keeps its hair. But we inject our killer cells alone. Those cells by themselves are enough to confer alopecia to the recipient mouse. This tells us that by killing these cells and only these cells, we have the chance at selective therapy and leaving everything else intact. So if we can find a way to do that, that seems to be at least one cell population that's important. So once we got the model working in the lab, we can use it in two different ways. First, we take one donor mouse and we take 10 little bits of skin from the back of a donor. We put it on a recipient, and then we wait 6 weeks for it to begin losing its hair on the belly, and 4 months it will lose its hair all over. These are our models for patchy alopecia and also for totalis universalis. So if we give a drug at the same time that we graft, we can measure and see if the drug actually prevents the onset of alopecia areata. That's an easy experiment. That's actually our lowest bar. So if a drug fails in that assay, it won't work in any other assay like reversal. But no patient comes in saying, I'm going to get alopecia 6 months from now. Please give me a JAK inhibitor. No. So the clinical context is this one, where the mouse is allowed to develop alopecia, and then we ask, can we give a drug either in early or late disease and actually get disease reversal and hair growth? So those are the different ways that you'll see that we've used the model. So again, with the bias in the field, the idea first was, can we take a biologic, can we take a blocking antibody for some of these critical pathways, such as interferon gamma or IL-15 or IL-2? Can we use those to block the onset of alopecia? And the answer is yes, we can block with anti-IL-15 or anti-interferon gamma. But was this really the most elegant way to do this for alopecia areata, right? We know that compared to, say, psoriasis or atopic dermatitis that are fortunate enough to have comorbidities that can maybe make them a little more tolerant to risk of taking a biologic, so far alopecia areata doesn't have that. So the safety margin for this disease is probably going to be much higher even than psoriasis or atopic derm. So the idea of using a strong biologic was not appealing to us, especially because of the need for injections, infusions, what have you. So this led my colleague, Raphael Kleins, the immunology colleague, to bring up the possibility that maybe we could accomplish the same thing using a small molecule JAK inhibitor, which you'll see here is downstream of IL-15, but also gives very nice selectivity because it gives us the chance to target particular JAK isoforms and not the entire signaling pathway as if we used one of those blocking antibodies. The other advantage, of course, is that these are small molecules, and the idea of being generated into a topical treatment was very appealing for alopecia areata, particularly for kids. And so very quickly, the JAK inhibitors became our focus for further treatment in the mice. And so again, the two main axes for alopecia are IL-15 signaling and interferon gamma signaling. This is the pathway that controls our cytotoxic T cells. This is the lighter fluid that controls the danger. You'll see that downstream of each of them, JAK1/3 is what's downstream of IL-15, and JAKs1 and 2 are downstream of interferon gamma. So they already share JAK1 as a common mediator, again, giving us some idea that that there might be an approach here that could block both arms of those important pathways. So when we began in about 2011, 2012, it was just before any of these drugs were approved. So Tofacitinib and Ruxolitinib are now both FDA approved for rheumatoid arthritis and for a group of different blood disorders for Ruxo. Baricitinib, I think, is now approved in Europe, but not quite yet here in the States for rheumatoid arthritis. Tofa is a pan-JAK inhibitor. It inhibits all three isoforms, JAK3 preferentially, then JAK1, and lastly JAK2. And Ruxo and Baricitinib are preferential JAK1 and 2, and they spare JAK3. Some of these others are in various other stages of development. Most of the work in the JAK field is on JAK2 because that's the oncology target that inhibits JAK2 in many different blood disorders. But for autoimmunity, the ones that really are under pursuit are JAK1 and JAK3. So the two that we had to start with really when we began were Ruxo and Tofa, and most of the work I'll show you has been on Tofacitinib. So again, coming back to the mouse, the first thing to do was to administer these drugs to the mice and see if we could at least, in the first case, prevent disease. So what I'll show first is systemic delivery of both inhibitors. These are given with a pump to the mice. So what you see here is that when we give Ruxo or Tofa, this one's Tofa, all of the mice in green here keep their hair, and all of the PBS treated mice, the control mice, lose their hair. You'll see this is a very nice, healthy looking anagen follicle. These are dystrophic anagen follicles that are infiltrated heavily with T cells. In this model, the lymph node and the spleen stay small because basically the killer T cells never get born because they've been given the JAK inhibitor from the time that we induce disease. So this is a prevention model. And the same thing works for ruxolitinib. So we can fully prevent the onset of alopecia areata by giving the JAK inhibitor at the time that we transfer disease. And so again, small lymph nodes, small spleen, and there's the infiltrate. So we can completely block the disease by doing it this way. When we compare that to the sledgehammer approach, this is a blocking antibody for IL-15. If you just look across the top row in each one, you'll see bright red staining. Those are our danger signals. It's our infiltrate, it's our MHC, it's our danger. And in each case, the lower row, you'll see that there's a lot less red staining and a lot more melanin here, suggesting that the hairs have gone back into anagen stage, whereas here they're arrested in telogen. So the JAK inhibitors work essentially just as well as the blocking antibody. The more important result is this one. So this is that 20% of skin T cells in untreated mice. This is by flow now. So this is anti-IL-15. They're still there. And these are the control mice for the 2 JAK inhibitors. This quadrant is after treatment. So the IL-15 blocking antibody as well as both JAK inhibitors can completely prevent that 20% of cells from being born. Important. So the question then became, that's nice, but again, this is the easy model. This is prevention. Can the same thing work if we try to do it with reversal? And so now we're going to switch both mode of delivery and the model. So now we're going to do topical delivery, and we're going to try to reverse mouse alopecia. So these are the control mice who were only given a vehicle, and that's before treatment, and then that's 1 month and 2 months. And these are mice that received both JAK inhibitors before and 1 month and 2 months, and the same thing. So you see robust regrowth here in the 2 treated mice compared to the control. Mouse skin is very thin. It's very easy to deliver just about anything to a mouse's skin. But again, the most important result for us is, are we reversing those signs? So here you see we are restoring our immune privileged environment in the hair follicle. So that was goal 1 by this red staining going away. And secondly, and most importantly, here you can see that even using topical delivery, again, it's in a mouse, we have essentially cleared that entire 20% of cells from the skin. That's extraordinary, that a drug that we can give topically can actually get rid of all 20% of those killer T cells. So this was very encouraging. It's a funny looking slide, I apologize, but it's 2 questions that we get all the time. The first question is, do we see systemic absorption? And the second question is, how long does it last? So these are mice that are photographed in a different way, but this is one mouse, this is looking down on its back, and this is flipping the mouse over, and here you're looking at its belly. So this is the same mouse with its back and its belly. This is after 12 weeks, back and belly, and that's 8 weeks after stopping drug, back and belly. Now if we do that for Ruxo, this is at baseline, so back and belly. At 12 weeks, the back, which is where we applied the drug, has fully regrown. You see nothing on the belly, so that tells us we're not getting a lot of systemic absorption for either of these drugs. And then for durability, if we just leave the mouse in the cage for a few months and do nothing, we keep the hair growth on the back, and we don't continue or grow any hair on the belly. And so at least in our very crude way of measuring this in the lab, and again, we're not professional formulators or professional mouse models of drug development. In our hands, we see good durability, and we do not see that the drugs travel to other parts in the body. So I'm not going to talk about this part today, but if you are in the audience and you have good eyes, you've probably realized that the mice that we treated topically grew their hair much faster, much thicker, and much darker than the systemically treated mice. And that was very striking to us over and over again. You know it's a good sign in the lab when I point something out and everybody in the lab laughs at me and they say, that's not true, that couldn't possibly be happening. So that's always heartening to me because it usually means I'm right, which never happens. But long story short, they did, my folks in the lab were brave enough to take that on, and it turns out that unexpectedly and completely by serendipity, the JAK inhibitors actually have a direct effect on activating hair follicle stem cells that's totally independent from their effect on dampening the activation response in T cells. And so when we give the drugs topically, we get the dual benefit of them turning on the hair cycle as well as turning off the T cells. And we've now launched a whole separate program on this in normal wild type hair follicles. But credit goes here to David Norris at Colorado, who often said to us, it's not going to be enough just to get rid of that infiltrate. You're going to have to do something else to turn on the hair cycle. And so again, my colleagues were far ahead of me. So that was the, excuse me, I'm just going to take a drink. That was the backdrop to the preclinical work. And with that, we were very lucky to be able to convince our clinical colleague, Julian Mackay, to begin to put these drugs into people. But before that, we really felt that it was going to be important to be able to measure the response to treatment in patients. And so we in the lab began in parallel to develop a series of biomarker signatures that from a patient's scalp biopsy, we could begin to quantitate their response to treatment. And I hope I can show you a couple of examples how this has become very useful. So this is just a series of biopsies from alopecia scalp. And if we just do RNA sequencing and we feed that data into the computer tells us immediately control scalp, patchy disease, and universalis totalis have very different signatures, and that there's also a gradient from normal to universalis totalis with patchy disease somewhere in the middle. Statistically, we were encouraged for our biomarker to choose not only genes that went up in disease, but also to choose some genes that went down. So in this case, we chose the hair signature, which are largely hair keratin genes, as well as our immune signature. So when we administer a treatment, it's kind of like a seesaw. These signatures should come down, and these signatures should start coming up. So we're monitoring kind of both ends of the treatment response. This is, again, totalis universalis. So we have very high immune signatures here of these two classes, very low hair signature because there aren't many hairs left here. And compare that to normal, we have very low inflammatory signature, a very high hair score because there's a lot of hairs in this biopsy. And in the middle, you see patchy alopecia areata, some immune signature and still some remaining hair. So when we scatter those statistically, they come out sort of along an axis that looks something like a 3D cube. So we borrowed a statistic from the lupus field, which they call the molecular distance to health. They basically collapse the entire thousands of data points in an RNA-seq experiment into a single data point, and it allows them to follow response to treatment by following essentially a single number. So black down here is normal scalp, patchy disease in the middle, and totalis universalis up here. So when we treat, what we want to see are the disease spheres from here migrating downward towards the normal range, and I'll show you some examples of how that works. This I just put in to, again, try and clear up a dogma issue. So we got an awful lot of feedback negative from this idea, but I think it's important to put out there just for your own thought. So the thinking in the field is that patients who have long-standing universalis totalis are essentially burned out, meaning that their infiltrates have left the building, there's no hairs there left to grow, that this is essentially a fire that's gone out a long time ago, and that these patients historically are more resistant to treatment, which we know is true. But our gene expression profiling told a very different story. You saw in that first graph that the patients who had universalis totalis had the most exaggerated dysregulated immune signature, not less, more. So this suggested that these patients still had a very active disease, but it was still very actively suppressing their hair follicles. But the corollary of that is it means that in our hands at least, and there's debate now in the field, even patients with long-standing disease, by this definition, should be treatable using these therapies. So again, there's different studies that show different things, but in our hands, even patients who've had disease for more than 30 years can respond. So, okay, so a bit about our study. So Julian Mackay, again, my colleague in clinical research, was very brave to start 2 open-label studies. The first one was Jakafi, or Ruxolitinib, and the second was Tofa, and I'll show you those. So our initial study was 12 patients with, again, long-standing disease in some cases. We went on to add 2 universalis totalis based on our early results. These patients got the approved dose of Ruxo, which is 20 BID, which is very high, but that was what was approved. And then we followed them essentially by SALT scores and by biopsies during the study. So in our study, roughly 9 out of 12 patients achieved our endpoint of at least 50% change in SALT score. The response is early. It's progressive. Once it starts, it keeps going. We have 3 complete non-responders, and we'll talk a bit about that. And all patients in all of our studies experience some degree of relapse, whether it's severe or back to baseline. A SALT score, as you all know, I think is a way to quantitate the residual hairs on the scalp and then count sort of in a quadrant manner how the hair regrows and put a number on that. So some examples. This is a patient at baseline. These are tattooed eyebrows that he's wearing here. Pretty extensive disease, duration of 12 years. This is him after 6 months. Full regrowth on the scalp, a beard, eyebrows, basically everything. This is another patient at baseline, and again, through his 5 months of treatment, he met our endpoint of regrowth in the middle, but I know you're already staring at his forehead because you see that it's repigmenting. So this is a patient that was sent to us by John Harris at Yale, who he also had treated for vitiligo. So it turns out that our same killer T cells are also the same cells active in vitiligo, and so this patient had a very nice dual response, and John went on to publish this with Julian, and shows here the quantitation of his vitiligo, and then after treatment showing that it relapses back to baseline here. Another subject after 6 months of treatment, again, almost a complete change. This is another subject. That's not our biopsy. Julian makes me say that. That's not our biopsy. That was there before. Full response. This is a non-responder, so it's a complete flatline. We don't yet understand the molecular basis of this, but in our hands at least, patients are very bimodal. If we can get them to respond at all and keep them on a high enough dose, they'll usually go to completion. If they don't respond at all at the beginning, there's a good chance that they're going to stay as non-responders. So in our initial study, essentially far less frequent adverse events than we thought we might see. One patient had a little bit of a lowered hemoglobin, adjusted the dose, and went back on treatment. Again, relapse is common in our hands, again, ranging from what's not noticeable to the patients to you would call back to baseline. These are the dropping SALT scores. So after treatment, the SALT score goes down. Remember, SALT score of 100 is universalis. So those go down, but it's easier, I think, more intuitive to view it as regrowth. So this is the opposite of the SALT score. And you can see there's the regrowth curves. The dashed lines are after stopping treatment. These are the patients relapsing here. And this is the non-responder lines down here, just completely no response. So what do we see below the surface? The more interesting part for us is we see histologically reversion of our danger signal back to baseline. We see at the RNA-seq level, this is normal scalp, on this side, so low immune signature, a lot of hair. At the beginning, they had high immune signatures and not much hair. And then after week 12, the immune signatures are essentially normalized to baseline. And here you can see the hairs are coming back. And that's shown here by the 2 red dots at baseline and the 2 green dots after treatment. When we expand that to the whole study, everybody between the yellow bars are the 9 responders. So at baseline, again, high immune signatures, not much hair. And then after treatment, we've normalized immune signature and the hairs are regrowing. Compare that to normal scalp here. So nice reversion on the immune side and regrowing hairs. But these are the 2 non-responders. And if you look at baseline, you don't see that same bright red immune signature. It should be there. But this tells us that essentially there was nothing there for the JAK inhibitor to hit. It didn't have this particular type of immune signature. It didn't have interferon gamma, didn't have killer T cells. We don't yet know what they do have because there doesn't seem to be a rhyme or reason to the non-responders, but we can at least say what they don't have. This suggests to us that if we introduced a screening test at the beginning, that a patient had to have an immune signature of a certain threshold in order to be a predicted responder, that might be a way to stratify patients into the use of a study or practice using a JAK inhibitor. If they don't have anything that matches that signature, chances are they won't respond. And again, that's an N of 2. It's not a huge number. We've had trouble getting non-responders to give us biopsies. They're usually not happy that they're the only non-responder in the waiting room, so not a lot of cooperation yet. So here's what it looks like. These are the responders at baseline, very high signatures, and these are the responders after treatment in purple. So you see nice movement of their scores. These are the non-responders at baseline, the blue ones, and these are the non-responders after treatment. No movement whatsoever, really showing that there's nothing there for the JAK inhibitor to hit. And so again, how we use that clinically, I'll show you in a minute, I hope. So this is the TOFA study, very similar design, again, long duration of disease in some cases. The design here was different. We started with the FDA-approved dose of 5 milligrams twice a day, and we had to escalate first to 15 and then up to 20 a day, 10 BID. Once we did that, we began to get responses, and now you've probably seen the literature in the field. Many places are starting with 10 BID right out of the box. So again, these are longer durations of treatment because of we had to wait 5 months for the IRB to approve the dose escalation. So these are shown as the time at the low dose, the time at the medium dose, and the time at the highest dose in order to get this response. So it's slightly different timeline. Again, longer, 36 weeks, but eventually a response. This is our only patient who responded fully at the low dose, and she didn't have to be escalated. If you look at the Stanford study, all of their patients were done on the low dose. The low dose is what you can get the Pfizer coupon for. So most studies are done open label with this treatment, and then once patients have a response, there's appeals made to their insurance to pick up the cost, and then usually they can escalate to the higher doses. Again, a nice full response here. Again, a non-responder, nothing whatsoever. These keep me up at night, to understand that. This is the kinetics. So you see people ask, does it come back as feathers? Do you see patches or clumps? I thought this was a nice illustration just to show it. So at week 4, even with your skilled eyes, if you only saw these, you might not call that yet a responder at week 4. But if she came in at week 8, yes, clearly. So we began to wonder if we could do things with our markers, even at this early stage of week 4. And I'll show you one thing that we've done. These are the SALT scores coming down. This is our good responder on the low dose. This is regrowth going up. Again, this is the one responder on the low dose. Everyone else required an escalation that took place about here. But it seems as though in our hands, and now in many of the other studies using this, that the higher doses are what are getting the responses. So this is a little trick that we're trying to use the markers, but I think, again, it may become useful. So this is the patient, again, at week 4. You might not call that a responder yet. But if you were giving a strong drug, an expensive drug like a JAK inhibitor, you might want to know, even at 4 weeks, am I seeing anything? Is she responding underneath the surface? And if you look here at 4 weeks, she's already had a 40% roughly improvement in her ALADIN score, in her biomarkers. So this tells us 2 things. 1, she's on the right drug, she's responding. And 2, that she's probably on the right dose because there's been movement even in 4 weeks, even though we can't see it yet visually in terms of regrowth. So she's probably on the right drug and the right dose. If we compare that to someone like this, at 4 weeks, she almost looks a little worse clinically. So can we do the same thing? So here, if we take a biopsy, you can see at baseline in 4 weeks, nothing, no movement at all, which reflects what we're seeing here. But if we increase her dose and let her go, she will have a complete response. So this tells us, 1, we don't know if she's on the right drug or the right dose, but if we increase the dose, we can then see it moving. So by using these markers early, we're hoping that we're going to be able to at least guide treatment a little bit to say, perhaps elevate the dose, wait another 2 months, and then call her a non-responder. In order to not do this by biopsy, now we've tried to convert this to a serum test where hopefully we can use a single marker available through a commercial lab that can give this result overnight so that you don't have to give your patient a biopsy and we don't have to do 6 weeks of RNA sequencing to get that result. So trying to make this more practical. And just to close this thought, Interestingly, in Pfizer's own studies on tofacitinib in psoriasis, they also got a marked differential in terms of response on 5 BID versus 10 BID, almost the same numbers that we're seeing in alopecia areata. So what does that say? Maybe you need a higher concentration so that it's going to get to the skin. JAK inhibitors have a very short half-life, so who knows? But at least what we saw in alopecia has been reflected in the literature in psoriasis as well. Every 2 years, the field, as small as we are, gathers in something called the Alopecia Areata Research Summit. It's coming up again this year in November. But 2 years ago was the first time that many of the folks presenting were beginning to use these drugs in their centers. And what I thought was amazing was that even though everyone was giving it different times, different doses, different patients, roughly speaking, almost across the board, the response rate hovers around 65%, 2/3 of patients, again, depending on which study you read and how they give it. It's a very consistent number. So that's also heartening to see it reproduced now around the world and in the literature. In terms of side effects, a couple of things have bubbled up. Acne, which again has been noticed in a few patient series. Weight gain, which the patients attribute to feeling happier and going out socially and having, just a better time socially being out in the world. They're very happy to keep their weight gain and their acne. So not a lot of patients, once they respond, are happy to come off. So this is just a quick rundown. There's been a huge explosion in the literature. And again, as a simple country geneticist sitting in my lab watching these things come over PubMed, it's extraordinarily gratifying to see now this work being tried and reproduced by others around the world. So it's an exciting time. So just in closing, just 2 last sections if I have time. Is that okay? So questions now are, what can we do better? Do we need to hit every JAK? Do we need to hit all 3 JAKs with tofacitinib? Can we begin to become more selective, more refined in our understanding? So we went back to our initial work and we realized that we didn't have a lot of evidence for JAK2. We have strong evidence for JAK1 and JAK3, as we knew, but we began to wonder, could we get rid of JAK2 inhibition and still keep our efficacy? The reason for that is because most of the side effects in the column of drop in platelets or hemoglobin are coming from the JAK2 axis. So if we didn't need to inhibit it, could we get away without it? So now there's a whole next generation of these inhibitors coming along that are in various stages of clinical trials. Most of these, luckily for us, we can purchase commercially, but basically we were able to get a collection of what we call JAK1 selective, JAK2 selective, and JAK3 selective, except for this one, which is a covalent JAK3 inhibitor. It's not selective, it's actually specific. So these mean that they're more selective on JAK1, but they still hit the others a little bit, but it's the best we can do. This one is a pure JAK3 by virtue of the fact that JAK3 has a particular cysteine at amino acid 909 that binds covalently to this drug. The others don't have that cysteine. So this one, and this is the one that Pfizer's presented their data most recently on, is in clinical trials by Pfizer. So when we put these in the mice, long story short, the JAK1 selectives give the expected response. The JAK3 selectives give the expected response. But in the pink box, you can see that the JAK2 selectives essentially don't work. So this gave us a nice indication that we could probably do away with inhibition of JAK2 and still not compromise our therapeutic effect. These are just the proofs of that, showing that in the skin, the JAK2 inhibitor looks like the control, whereas JAK1 and 3 inhibitors are getting rid of our inflammatory markers. Here by flow sorting, the same thing, I won't belabor it. The same thing here, we're getting rid of our T cells with the JAK1 and the JAK3 inhibitors, but not the JAK2. So the long story short there is that we've begun to get at a mechanism for understanding this, and in addition to JAK2 being dispensable, we also have evidence now that the JAK1/3 inhibitors may actually deliberately be inducing a state of T cell exhaustion. Basically, they're putting those T cells into a twilight sleep, if you will, selectively, so that when we withdraw the drug and they wake up, this may give us a handle on relapse. And so if we can understand the mechanism by which the inhibitors are putting the cells to sleep, we may have a way to monitor that sleep state, if you will, so that we can perhaps predict flares later on. So maybe giving tools to help you when you're using these drugs in the future, how to tweak them. And we've also shown that the same is true topically. So I mentioned at the beginning that there were only 2 approved JAK inhibitors. There's actually a third. It's a veterinary JAK inhibitor called Apoquel for itchy dogs. So Apoquel is actually the perfect profile. It's a JAK1/3 inhibitor that apparently saves many dog owners much heartbreak of their itchy dogs getting better. It's constantly back ordered. It's like the most popular itchy dog drug that there is. Interestingly enough, for those of you that have used Ruxo and Tofa, you know that it ranges from $5,000 to $10,000 a month. You can cure your dog's itch for far less than that. So this is our little mouse hospital. So everything on the left is how we move these drugs from prevention to treatment to topical and then into humans. You can see not too many have made it across, but we're hopeful that this will continue and some will. Again, I won't belabor, but just to say that we've also thrown all of the known psoriasis drugs and Dupixent and any of the new biologics at the mouse model, and essentially nothing in our hands works in this class of drugs as we've seen in the human patients. But up here, we're constantly expanding our cabinet of JAK inhibitors just to, again, get a better handle on how they work. Last 2 minutes? Okay, so this is a little gross, so bear with me. So we've been interested in a long time in understanding the triggers for disease. Why does the disease start in one family member but not another? Why does it flare? Why does it come and go? Why do patients report after a stress? We don't know, but along with most of the rest of the autoimmune field, we wanted to ask whether the microbiome actually plays a role in the onset of alopecia areata. So why would we study this? With the idea, hopefully, of using a very low-tech way of changing the course of disease, potentially by manipulating the microbiome. So the first experiment that one does when you want to ask this question is you basically take your mouse and you hit it with a 4 antibiotic cocktail that you see here that, again, will kill everything in a 10-block radius. And then you ask if there's any impact on the course of your disease. We expected to see no impact on the course of our disease, but we were fooled. And it turns out when you treat the mice with this cocktail of antibiotics, you completely prevent the development of alopecia areata, completely. So we've done this many, many times. We've taken some of the antibiotics, put them in, take them out. Doesn't matter. This is a very reproducible result. The question for us was, is it their skin microbiome or their gut microbiome or potentially both? What are we actually affecting? So we can do this now all by sequencing. The first thing we do is sequence the skin microbiome. So we take swabs of the mouse skin and we pluck some of their hairs. And long story short, there's basically no change in the skin microbiome using this cocktail. But the question then was, is it coming from the gut? So the poop emoji is greatly helpful when you're talking about this. So what we do is pick up the droppings in the cage of the mice, and then we send them for 16S bacterial sequencing. So what we do is compare the affected versus the unaffected mice, and you can see here, in contrast to the skin microbiome, we do see a massive difference in the gut microbiome in treated, antibiotic-treated versus untreated mice, suggesting that this effect is coming from the gut. So the whole half of the lab is now figuring out what are those specific bugs in the gut, et cetera, in the mice. Another group in the lab has taken the bold leap to say, well, what do we see in patients? And so we've now done a skin and gut analysis on 10 new-onset alopecia areata. So these are naive to treatment on purpose. So again, the skin microbiome shows no difference. All these overlapping circles means there's no statistically significant difference in skin. However, in gut, you can see these 3 spots here where there is a significant difference in the gut microbiome of these patients. And when we feed these to the computer, remarkably, and again a shock for us, the computer clusters, with one exception, all of the control patients on one side and all of the alopecia patients together. This red and this red is the dysbiosis, the change in the microbiome in the alopecia patients, versus this, which is in the controls. Again, 10 people walking down the street in New York with new-onset alopecia was a bit of a shock to us. Again, we were strong disbelievers until we got the sequencing back and we saw this. And so it turns out that the change in the gut microbiome in alopecia patients is twofold. One is there's an overrepresentation of certain species of Firmicutes, in particular certain Clostridia, the healthy ones, not C. diff. And there's a down representation, underrepresentation of certain Bacteroides. You would say, okay, well, what does that mean, right? Why would it be those two classes? And again, when we turn to the literature, it turns out that the dysbiosis of these two classes of bugs are widely reported in IBD, in lupus, in multiple sclerosis, even on other models in a simple Western diet. So again, it's an N of 10. Take it with a grain of salt, but it is a first clue, at least, that there's a quite significant dysbiosis in alopecia patients. That was going on in the lab at the same time that this case report came out last fall. These are two patients, one patient here, this is a patient, and another patient in the paper, who both were treated for C. diff infection with, again, I'm sorry, a fecal microbiome transplant, which means that they have a colonoscopy prep and then they are repopulated with donor fecal material that either comes from a household control or from a bank. There's a bank that supplies screened fecal microbiome. And in both patients, they had complete regrowth of their hair and also durable. So these are up to 2 years. So we were, of course, ecstatic seeing this in literature because it made our crazy experiment seem a little less crazy. And we're now launching both a pediatric and an adult FMT study. Turns out we have experts in FMT at Columbia, so who were delighted to help us with this. So stay tuned, but I just wanted to share that as kind of an alternate pathway that's under pursuit. So I'm just going to close by saying it's been a wild year for JAK inhibitors in alopecia. This was 2017. This is 2018. The field is getting very, very active. And I have to say that both as a scientist and as a patient, I must say, this is monumental because it's the first time historically that any drug company, let alone 6 drug companies, have undertaken trials in alopecia areata for its own sake. That's important, not as a one-off of psoriasis or atopic derm or anything else. And that to me is enormously rewarding. So I hope I've convinced you that functional genomics can work when you make a good map and you kind of open your mind to where the genetics will take you. It can lead you on unexpected journeys and hopefully someday to a new way of thinking and treating the disease. A huge thank you to my team. Below is the sort of basic science that underlies everything we do. On top is the clinical translational, and a huge thanks to NIAMS who awarded us a P50 Center grant to continue this work 2 years ago. Without that, it would be tough. And a thanks again to the lab and to old colleagues and new colleagues for all of their enormous contributions, and that's the team. Thank you very much.
With the TOFA inhibitors, did you see any relapse when you took those patients off those drugs?
Yeah, so we see relapse in all patients, really, usually to greater or lesser degrees. Most patients go back to baseline. Occasionally, a patient gets worse than baseline. Sometimes they just report increased shedding that isn't quite visible clinically, but essentially everyone has some degree of relapse. The bar has become unexpectedly high. People say now, well, can't you cure us? Why can't we just take this for a while? Well, sure we could if we did a bone marrow transplant. I'm sure we could if we got rid of your evil T-cell clone altogether. But the thinking is no different than psoriasis or atopic derm. It's management. It's keeping the immune response low enough and controlled enough that you prevent breakthrough, but it's probably going to be lifelong treatment, just like the rest of what we deal with. So somehow the bar got moved very high in the past 2 years. I don't know why, but I think the enthusiasm needs to be put in the context of risk-benefit, first of all. These drugs have side effects. There's no question about that. And secondly is how they're going to be used. Maybe the use will be 3-4 months of a high dose of oral and then transition to a topical for maintenance, just like you do with everything else. So no different, really. And I hope the companies will, I think they'll be keeping that in mind, because there's development of both orals and a few topicals now. So I think management might not be unlike the way you all use steroids or other things. So I don't know how the bar moved, but it needs to be. We don't talk about cure. We talk about management the same way as everything else. If we get really smart and we can barcode those T cells and know what the antigen is and selectively kill them that way, that'd be nice. But so far, the thinking really should be very similar to how you handle the other diseases, except not with biologics, but with small molecules. Yeah.
Thank you so much for your talk. I've heard you speak several times. Every time I learn new things, so I really, really appreciate it. I'm a pediatric dermatologist, and this really hits home. Something that was interesting is that I have a patient who has Totalis and has had it for years. We tried methotrexate, we tried prednisone, and eventually we came to terms with this is not working, and we're going to just approach this psychosocially. She has a good support system. She's been to camp, etc. Her mom just emailed me. We picked her up from camp, and she had peach fuzz, and now she has a complete regrowth of her hair with nothing. So, of course, we were extremely excited. So I wonder if it would be interesting if we could capture those people. Why is it 5 to 8 years later?
Environment. It's the camp microbiome.
That's what I was wondering. So it would be so cool to just be able to quantify that and then
sequence the camp. When no, seriously.
Well, it is the AAD camp, so
no,
that's not implausible.
You hear about these clusters. Why are 8 kids in a single school coming down with alopecia? Everybody's thought about environmental toxicants. Probably the cafeteria. Probably the cafeteria. So a lot.
Well, I can't wait till you publish those papers. Thank you.
Maybe we'll send it to you as a reviewer. So far, it's been a little tough. Sorry. Thank you. By the way, if you didn't ask, but Tofa is about to be approved for juvenile RA down to age 2, and both the Lilly study and the Pfizer study are admitting patients as from 12 to 65, not 18 to 65. So if you have adolescents that are interested. For both of those oral studies, and they're both enrolling 700 to 800 patients, so they're large. So if you have teenagers that are interested, both of those studies are open to adolescents now. Yeah, sorry. Yes.
Hi, I'm Christine DeWitt from Georgetown.
Hi.
And I just want to thank you so much. I love this topic so very much. I have several patients who are on Tofacitinib, and I can actually confirm also the response that we've had with a clinical degree of certainty. We've also seen some of our young adults. I've had 2 cases of zoster develop. So I definitely agree with that.
Yeah, we've seen that too.
I have a couple of questions and a couple of comments. Number one, were you able to see if the CD8 T cells were present in humans to the same percentage as in the mouse models? And then in the responders, the human responders to TOFA, were the non-responsive patches biopsied, and did they have different expression markers from the patches that did respond?
I'll take the middle part first. So our paper just got accepted to JCI, showing the results of the human TOFA data, including T cell receptor sequencing. I don't actually know if it's online yet. So if it's not, it will be soon, and that has the detailed profiling of all 12 of the TOFA patients. Unfortunately, there was only one real non-responder in there, so we don't have great non-responder data. But your other question about was a patch rebiopsied after treatment or before, we actually prevented ourselves from doing that because we did the earlier experiment of biopsying and sequencing untreated patients, lesional and non-lesional. And you would think that non-lesional alopecia scalp would look more like control scalp. It clusters almost the same as lesional scalp. So when you look at 2 different sites, affected and unaffected, on a single patient, their unaffected scalp is essentially poised to fall out. All the markers are up. Everything looks like it just needs a little bit of a push. I would say that's the majority. We have other patients whose untreated, unaffected scalp looks more like control. So it's essentially all over the map. We felt on the basis of that it wasn't going to be instructive, and the patients at that point were reluctant to biopsy during treatment of a non-responsive patch. And as you saw by the regrowth, there were really seldom non-responsive patches because they kind of regrew uniformly, at least in our initial study. So we didn't do it mostly because of the non-lesional data, which isn't in the paper because we don't know what to make of it yet. So anyway, if you're interested, you can look up the new paper, and most of what you asked will be in there. But we have sequenced and we have quantitated CD8 response in humans versus circulating CD8 responses, and it's different from the mice. There's clearly more of a CD4, a mixed CD4-CD8 response in the humans that's different from the mice. So we just need more, 800 patients would probably tell us what we need to know, but it's only 10 or 12, so.
And one more comment is I'm really interested to see what happens with the microbiome. I've independently had several patients approach me about doing fecal transplants on their own, and I've done some research into commercially, air quote, available DIY. So basically the idea is that family members would send in fecal samples, a company lyophilizes the samples and then puts them into these capsules. The risk with that is that the capsules have to be of a specific material because otherwise there have been risks of small bowel bacterial overgrowth, so it's not getting to the place that it needs to be. So I've been counseling patients just to hold their horses.
Yeah, so what you just described is the selection of the donor is critical, right? So most of the work that's been done in that context has been for C. diff. And in C. diff, it's actually okay to use a household control. It's preferred because chances are the only thing that's different, more or less, between a household control and the patient is the C. diff. If you're looking to really cure the environment for alopecia areata, you absolutely don't want somebody from the household because their microbiome is probably very similar. It's just that their genetics are different and they're not susceptible. So for our study, we're actually purchasing it from a bank called Open Biome, which guarantees that the person had no autoimmune disease, but it's also been screened for everything. The other thing that we're going to do in our study is use the same donor for all of our recipients so that we can actually follow, and hopefully it's a good donor, hopefully follow, at least in a small, maybe 6 of the first 12 patients, follow the microbiome as because it drifts back to whatever it was. So, so it's very important that you think about what you're trying to achieve when you use the source of the donor. Household control is probably not the right control for this. And there's there's only a few studies on Crohn's actually where they've used, donors and they talk about the selection, but that's that's why we sort of went far outside the house. So it's probably wrong, but we'll see. So it was a start.
Hi, I'm Arjun, one of the medical students. I just had a question. I liked the end of your talk on the microbiome. I was wondering if there's any thoughts on how the gut microbiome versus the skin microbiome is affecting the immune response in alopecia areata, and if it would be worth even looking into seeing if there are any changes in the biomarkers that you talked about in patients where you have that fecal transplant. Is the microbiome change affecting the ALADIN markers?
You clearly read our next grant.
Oh.
So we've taken 2 lessons. One lesson is from Crohn's disease to answer the question about the leaky gut. In Crohn's disease, the model is that there's some changes in the gut epithelium itself that leads to increased permeability so that bugs that are there and present in the gut are somehow leaking and becoming more exposed. Not sure I buy it. The second model that we're following is actually a model developed here at the NIH on uveitis. And it turns out that uveitis is actually primed their T cells in the gut under the influence of the microbiome, and they attack at a distant site that's immune privileged. So we think that uveitis may be a good sort of model for us to pattern what we're doing in terms of the priming step. The other source that we're keeping an eye on, and I think this has become a hobby of mine, but I think there's a lesson there for this. If you look in the cancer immunotherapy literature recently, you'll see that the efficacy of of pembrolizumab and nivolumab and ipilimumab are dependent on the patient's microbiome. It's the same two classes of bugs that are dysregulated in autoimmunity that are dysregulated in cancer immunotherapy. So my hunch is that because we're trying to do the same thing, right? So in reverse, they're trying to draw T cells into their organs. We're trying to keep T cells out of our organs. I think there's a lesson there too. I don't know what it is yet, but we don't have a great roadmap, but those are the three fields that we're kind of tracking right now. One of our GWAS genes is actually expressed in the gut epithelium as well as the hair follicle. So that's become an interesting candidate for now. Not a great answer, I'm sorry, but
No,
that's
that's
where
great.
we're thinking.
Thank you.
Sure.
Maybe one more question. All right, great. Thank you so much.
Thank you for having me.